Now coming to sterilization and disinfection. Sterilization is destroying or killing all the microorganisms, making an article free from all the pathogens, non-pathogens also, including the spores. So basically killing all the bacteria with spores as sterilization. Disinfection is killing or disinfecting everything except the spores. So here spores are not killed in case of disinfection, and that is the differentiating feature between sterilization and disinfection. So in sterilization, destroying or killing all the microorganisms plus their spores. Coming to the classification of sterilization, dry heat and moist heat. In dry heat, you can use the flame, that is flaming, incineration, that is we do for hospital waste, we burn it, and hot air oven, we do it 160 degrees Celsius for 2 hours. We use the hot air oven for glass material, glass particles and all. Moist heat sterilization is using heat as well as the pressure or moist. So it can be at 100 degrees Celsius, less than 100 and more than 100. So at less than 100 degrees Celsius, we do for the pasteurization of the milk. The milk you get in the packet, this is a pasteurized milk. If you use unpasteurized milk, you are more prone to develop the tuberculosis, and that is why we use pasteurized milk or boiled milk. So pasteurization of the milk is done using two methods. Either you can use the flash method. Flash method is the best method for the pasteurization of the milk, most effective method for the pasteurization of the milk. Again, this is a important point that you need to remember. Flash method is the most effective method for the pasteurization of the milk. In the flash method, we use 72 degrees Celsius for 15 to 20 seconds only. And that is why it is the fastest method that is used for the milk, that is the packed milk. Holder method, you use 63 degrees Celsius for 30 minutes, that is half an hour. It is a quite longer process to pasteurize. Inspissation is done at 80 to 85 degrees Celsius, that is done for 20 to 30 minutes, and it is done for 3 days. Or you can use water bath, that is less than 100 degrees Celsius. At 100 degrees Celsius, we do tyndallization, that is 100 degrees Celsius for 20 to 30 minutes, and again for 3 days. For 3 days. Boiling at 100 degrees is again a moist heat at 100 degrees. More than 100 degrees is the autoclave, 121 degrees Celsius for 15 minutes for 15 psi per square inch. So the sterilization cycle, this was the question that was asked in NEET 2018 exam. What is the sterilization cycle? It is 121 degrees Celsius, 15 per square inch pressure, and for 15 minutes. This is a sterilization cycle, very important point that was asked in NEET 2018 exam. Now dry heat and moist heat. Dry heat kills due to protein denaturation. It breaks the protein of a bacteria. Also does the oxidative damage and the toxin effect of the elevated level of electrolytes, and therefore it kills the microorganism. Moist heat does by denaturation of the proteins as well as coagulation of the protein. So this is the difference between killing by dry heat and killing by moist heat. Flash method is the best method or better method for the pasteurization of the milk, we already talked about. And filtration is done for heat sensitive substances. For example, if you want to sterilize, or if you want to do the sterilization of a serum, sterilization of a vaccine, so we use this filtration method for the filtration of serum or vaccine. This is used for heat sensitive substances. Membrane filtration is best to sterilize the heat labile solutions. So membrane filtration is the best to sterilize the heat labile solutions. Most commonly used filter is this membrane filter. Membrane filters are the most commonly used filters, and these membrane filters are made up of polyester. They are made up of polycarbonates and cellulose ester. So remember, vaccines and serums are sterilized by membrane filters, by filtration method. So filtration method is used for the serums and vaccines, heat sensitive substance and heat labile solutions by membrane filter. And membrane filter is made up of polyester, polycarbonate and cellulose esters. Now tyndallization. The instrument that is used for tyndallization is Koch and Arnold steam sterilizer. So tyndallization is the method of sterilizing using the moist heat at 100 degrees Celsius. Then total or plate count or Ressa's urine test or methylene blue reduction test. These tests are done for to see or to test the bacterial contamination in the raw milk. Pasteurization. Pasteurization, the best method is the flash method. We already talked about. So pasteurization is done to check the milk-borne infection, and in flash method, you use moist heat at 72 degrees Celsius for 15 seconds and rapidly cool it. So 72 degrees Celsius at 15 seconds followed by a rapid cooling for the flash method, which is the most effective method for the pasteurization of the milk. And pasteurization of the milk is done for milk-borne infections. Coxiella burnetii, it is resistant to moist heat and it can survive the Holder method, and that is why we use flash method. Flash method is better than the Holder method. Then pasteurization milk is tested commonly by the phosphatase test. Phosphatase test. Culture medias are sterilized. Okay, pasteurization does not kill the endospores. Culture media are sterilized by autoclave. The essential parameters for steam sterilization. So it is for steam, that means autoclaving, we are talking about time, temperature, and saturated steam and pressure. So these are the essential parameter. What is the sterilization cycle? 121 degrees Celsius, 15 psi pressure, and 15 minutes. If the bioburden is too high, the established sterilization parameters may not be adequate, rendering the sterilization process ineffective. The most effective method of sterilization by the heat is autoclave, which is steam under pressure, or you can say it is a moist heat sterilization. So the best method, if they ask best method of sterilization or the most effective method of sterilization, it is steam under pressure. Steam under pressure is what? It is autoclave. What is the autoclave sterilization cycle? 121 degrees Celsius, 15 psi pressure for 15 minutes. Moist heat kills the microorganism by denaturation and coagulation of the protein, and dry heat kills by denaturation and oxidative damage. We have already talked this. For hot air oven, the holding time is 160 degrees Celsius for 2 hours or 1 hour. Now, very important point is plasma sterilization. In NEET 2019, question was asked from plasma sterilization. It is the newer method of sterilization. In this, hydrogen peroxide gas is used. So plasma sterilization uses hydrogen peroxide gas. What happens? From H7O7 plasma radicals are produced. So the sterilization controlled is tested by the spores of Bacillus stereothermophilus. Gas plasma sterilization, it uses what? It uses hydrogen peroxide gas and radio frequency emissions. And it is tested by the spores of Bacillus stereothermophilus. Again, the gas sterilization is tested by the spores of Bacillus stereothermophilus. Coming to chemical sterilization, we use phenol, which is carbolic acid. Isopropyl alcohol is better than the ethyl alcohol as it is a better fat solvent. If 100% concentrated alcohol is used, it can cause dehydration of the skin proteins, and that is why we do not use. Aldehyde. In the aldehyde, we have formaldehyde. Formaldehyde can be used in two forms. It can be used in gas forms, such as in fumigating the OT. It can be used in liquid form as the specimen preservation. We use formaldehyde for the specimen preservation. Glutaraldehyde 2% is Sidex, which we use. So glutaraldehyde is used for all of the following except glutaraldehyde is used for bronchoscope, glutaraldehyde is used for proctoscope, glutaraldehyde, this is for anal examination, this is for lungs, broncho, endoscopy, endoscopic tube. So glutaraldehyde, 2% glutaraldehyde is what? It is Sidex. It is not used for thermometer. Best method of sterilization for fiber optic laryngeoscope, it is glutaraldehyde. Again, as I told you, glutaraldehyde is used for all the things that goes inside, either a bronchoscope, a laryngeoscope, a proctoscope, a endoscope, all the scopies, we use the glutaraldehyde. And 2% glutaraldehyde as a disinfection is what? It is Sidex. So concentration of Sidex used as the disinfection, it is 2%. Cystoscope, bronchoscope, all the scopes are disinfected by glutaraldehyde. Either 2% glutaraldehyde is given in the option, or they can also give Sidex in the option. So you have to see what is given in the option, glutaraldehyde or Sidex or 2% glutaraldehyde, all are the correct options. Dettol is 4% chlorozinol, oxinol. Savlon contains cetramide 3% and chlorhexidine 0.3%. Very easy. Soaps are anionic surface acting agents. Now coming to halogens. Halogen we have chloride and iodine. So chloride we use 1%, sodium hypochlorite. Now sodium hypochlorite we have also talked about in endodontics. So we use sodium hypochlorite as irrigant. Here 1% sodium hypochlorite is used for blood spills. Any blood spill is washed with this 1% sodium hypochlorite. So blood flow on the OT is cleaned by, this was the question asked in AIMS, blood flow on the OT is cleaned by the chloride based compound, or they can give sodium hypochlorite, 1% sodium hypochlorite. So best answer is 1% sodium hypochlorite. 1% sodium hypochlorite is used to clean the blood spills. The blood that is spilled over the floor, that is spilled over the surfaces is disinfected or cleaned with 1% sodium hypochlorite. Sodium hypochlorite is not used as disinfectant for any skin preparation because it is an irritant. And remember, when we talked about sodium hypochlorite in the endodontics, we use rubber dam because it is highly irritant to skin. That is why while we are irrigating the canal with sodium hypochlorite, we use the suction along with that, because whenever it touches any of the surface, it causes irritation of the mucosa also. So it is not used as skin disinfectant. It is used for washing this floor and all the blood spills on the floor. So hypochlorite is not used as disinfectant. And this disinfectant for skin preparation, this was a question asked in NEET 2018 and 19 exam because it has got the irritational property. Concentration of the Lysol that is used as disinfectant is 3%, and concentration of the phenol that is used as disinfectant is 5%. Again, very important, ethylene oxide, ETO gas. It is basically a gas sterilant, and we have two cycles. The cold cycle is 37 degrees Celsius to minus 5 degrees Celsius, and warm cycle is much higher, 54 degrees Celsius to minus 5 degrees Celsius. So basically this ethylene oxide, it is a gas. It is an alkylating agent, and this has got a highly penetrating power. This gas is very penetrating. It is also explosive, and it is used for to sterilize this pre-packed syringes and all. So ethylene oxide, important point, is alkylating agent. It is highly penetrating power. It is explosive, and it is used as the pre-packed syringes are better sterilized with alkylating agent than the gamma radiation, also used for ICU equipments. And to control, to check the ethylene ETO gas, we use the Bacillus globigi. These are the spores that we use, spores of Bacillus globigi, and these are the spores that are used to check the sterilization. So heat labile instrument that are used in the surgical procedure. So basically ICU equipments can be used with ethylene oxide gas. Biological indicator for ethylene oxide is the spores of Bacillus globigi. Ethylene oxide is effective for sterilizing the mattresses, sutures, and the plastic petri dishes. Ethylene oxide sterilized products requires 8 hours of aeration. Ethylene oxide is a highly toxic agent. It is also explosive. It is highly alkylating agent, and it destroys the microorganism by the process of alkylation. Now coming to the Sploding classification, in that we have non-critical devices, we have semi-critical devices, and we have critical devices. So non-critical devices require low level of disinfection. These are non-critical. As the name suggests, these devices are not critical. So they require low level of disinfection. Even if you do low level of disinfection, you can use. So they comes in contact with the intact skin. For example, you are using BP cuff or stethoscope. They require very low level of disinfection. So these are non-critical devices. Semi-critical devices comes in contact with the mucous membrane. For example, our mouth mirrors, probes, or in medical like endoscope, laryngoscope, so they require high level of disinfection because it is coming in contacting to the mucosa. Then we have the critical devices that enters the surgical site. So after when you are doing any operation, say any surgical instrument or any implant or any dental instrument, these are the critical instruments that require the high level of sterility, that require the high level of disinfection, which enters the surgical site. So what is high level disinfection? What is low level disinfection? What is intermediate level disinfection? High level disinfection, it kills the vegetative microorganism, it inactivates the viruses, but not necessarily the high number of spores. But though this high level disinfection, they are used relatively for short period of time, for example, glutaraldehyde. The intermediate level, it kills the vegetative microorganism, including TB, fungi, and inactivates most of the viruses. Most, not all the viruses. Here in high level, we have inactivation of the viruses. Here, most of the viruses. The chemical germicides used in this procedure are often correspond to the Environmental Protection Agency that is approved by hospital disinfection. So TB, very importantly, TB is also killed with this intermediate level disinfection. Then low level, the procedure kills most vegetative bacteria except the tuberculosis. Here in low level, tuberculosis is not disinfected, it is not killed, some fungi, and it inactivates the enveloped viruses. So they are the hospital disinfectant or sanitizers. Now coming to the culture media, it is sterilized by autoclave. Sharp instruments, you can use 5% cresol. Also, you can use hot air oven. Best option is 5% cresol. Thermometer. When you remember, I've asked this question, glutaraldehyde is used for all except questions. So thermometer was not the answer, it was the exception. So glutaraldehyde cannot be used to sterilize or disinfect the thermometer. Then what can we use to disinfect the thermometer? It is Savlon. Cystoscope, all the scopes, cystoscope, bronchoscope, laryngoscope, endoscope, all these scopes can be sterilized or disinfected by 2% glutaraldehyde, which is also known as Sidex. Now, how do we check the sterility? What is the test organism for dry heat? For dry heat, we use the non-toxogenic strains. Remember, non-toxogenic strains of the tetanus. So, Clostridium tetani. These are the non-spores. We use spores for everything. So spores of non-toxogenic. This is not going to cause the tetani. Then for moist heat, we use the spores of Bacillus stereothermophilus. And also Bacillus stereothermophilus is used for plasma sterilization, plasma gas sterilization, in which we use hydrogen peroxide gas. And Bacillus if you remember, Bacillus globigi is used for the ethylene oxide. So plasma gas, Bacillus stereothermophilus, autoclave, Bacillus stereothermophilus, and Bacillus globigi for the ethylene oxide gas. Then we have Riedel-Walker test or phenol coefficient test. In this we use Salmonella typhi bacilli. The organism most resistant to sterilization is prions. Now, the important point they ask from the test organism is biological indicator of the autoclave, which is the moist heat sterilization, which is sterilization steam under pressure. So the biological indicator for the autoclave is Bacillus stereothermophilus. And the spores used for analyzing the efficacy of the autoclave is again the Bacillus stereothermophilus. Now coming to the materials and the method we use to sterilize. All the suture material except catgut we use autoclave. Catgut, for catgut we use the ionizing radiation. We use the ionizing radiation. Rubber gloves, surgical instrument, and most of the culture media. We already talked about for culture media we use the autoclave again. Forceps, scalpel, scissor, needle. All these are the pointy surgical instrument. For these we use the hot air oven. Disposable syringes, we use the ethylene oxide gas. And suture materials, for suture materials we use the autoclave except the catgut. For catgut we use the ionizing radiation. Hospital waste, incinerate. We burn all the hospital waste, incineration. Vaccine serum antibiotic solutions, filtration. We already talked about filtration. Then OT is fumigated with the formaldehyde. So formaldehyde, we have gas, and the liquid we use for preservation of the specimens, and gas we use to fumigate the OT. Preparation of the skin for surgery. So when you are preparing any skin for the surgery, you use the iodine spirit, Savlon, or the chlorhexidine. So all these things can be used. For paper points, for brooches, files, or any root canal instrument, we use glass bead sterilizer. Also you can use the also you can say it the hot salt sterilizer or the glass bead sterilizer. Both are one and the same thing. For cotton pliers and cement spatulas, you can use the flames, so you can pass it through the flames several times. For dampened dishes and for glass slab, you can use the thermosol, that is followed by the double swabbing it with the alcohol. Then GP points, gutta percha. We already talked about this in endodontics. For GP, you use 5.2% sodium hypochlorite for 1 minute. First, you dip this in 5.2% sodium hypochlorite for 1 minute, and then you rinse it with the hydrogen peroxide, and then you dry it. For silver cones, you pass it through the Bunsen burner flame several times. And LJ medium, you use the inspissation. Inspissator is used. So LJ medium is used for tuberculosis. Sugar medium, we use sterilization technique or intermittent sterilization. The best way to dispose the hospital soiled dressing, human pathological material, and animal carcasses. So basically this is to dispose the hospital waste. To dispose the hospital waste, we use the incineration. Incineration is just burning it, dumping everything and burn it. Volume reduction of the infectious hospital waste is 85%. Best method of sterilization of disposable gloves and syringe is ionizing radiation, or you can say gamma rays. Oil, dusting powder, and grease are sterilized by the hot air oven. Best method of sterilizing the metallic instrument is again hot air oven. Glass pipettes and flasks are sterilized with again dry heat sterilization, which is hot air oven. Culture media that contains eggs or serum, very important point, is typically sterilized with inspissation, which is moist heat at 85 degrees Celsius. Now Brownies tube number 1 is used for steam sterilization efficacy and use of gamma rays, that is cold sterilization. In gamma radiation, the radioactive isotope you used is cobalt 60. Thermal death point. Thermal death point is the lowest temperature that kills all the microorganisms in 10 minutes. So the lowest temperature that kills a bacteria under the standard conditions in a given time is known as this thermal death point. So this is the lowest temperature that kills a bacteria. Under a moist condition, for most of the vegetative microorganism, mesocephalic bacteria, they have a thermal death point between 50 to 65 degrees Celsius, and most spores between 100 to 120 degrees Celsius. And that is why we use 121 degrees Celsius in the autoclave, because we kill the spores in autoclave.